So welcome everybody to the second webinar of the VALITEST webinar series on the organization of test performance studies. So VALITEST is an EU-funded project built to improve the reliability of diagnostic tests performed in plantless laboratories across the European and Mediterranean region. The project is undertaken by a consortium of 16 partners composed of research institutions, private companies, national plant protection organizations, and one intergovernmental organization. So you can find more information on the VALITEST project on our website. Test performance studies are inter-laboratory comparison that aims at evaluating the performance of diagnostic tests. And during this series, the VALITEST partners will present the different steps of the TPS workflow through different videos, webinars, and training activities. So we had our first webinars last week, and also the first videos were released. Today, we will present the different elements that should be considered when selecting the tests to be included in a TPS. So this presentation will be recorded, and a link to the recording and the PDF presentation will be provided on the VALITEST website. Today, our speaker is Anna Moisiz. So Anna is a member of the VALITEST coordination team. She joined the plant health laboratory of ANSES in France in November 2019 to manage the VALITEST project. She has a PhD in chemistry, and she worked on research projects for more than 10 years, particularly with the diagnostic company in human health, bioMérieux. And then she developed experience in quality management, working for 7 years in pharmacy, food, and agriculture-related industries. So we want to acknowledge that Géraldine Antoine, which is the VALITEST project coordinator, could not join today's webinar. And this is why Anna is replacing her to make this talk on the selection of tests. So Anna did not organize TPS, but she was involved in the management of the documentation produced in the framework of VALITEST TPS organization. So at the end of the webinar, there will be a Q&A session, and 3 TPS organizers will join us, and you will be able to ask all the technical questions you want to these TPS organizers. So this is Anne-Marie Chappé from ANSES in France, who organized the TPS on Bursaphenancus xylophilus. Marta Lucci, who works for CREA in Italy and organized the TPS on TOB-RFV, and Tania Dreo, who works at the National Institute for Biology in Slovenia and who organized the TPS on Erwinia amylovora and Pantoea stewarti. So this is all for the introduction, and with no further ado, I will give the floor to Anna.
Thank you for the introduction, Charlotte. So for this webinar, the selection of a test and associated document, I will first make an introduction on TPS workflow and definition, and then I will present you the common rules defined in the framework of VALITEST project. That will be illustrated by data from TPS organizers, by one example related to the TPS organized by Anne-Marie Chappé on Bursaphenancus xylophilus, and by interviews Charlotte realized with TPS organizers. At the end, like Charlotte mentioned, there will have a question and answer session, and I would like to thank Anne-Marie Chappé, Marta Luigi, and Tania Dreo to be present today to answer the practical question that you might have at the end of this webinar. So the TPS workflow corresponds to a few steps from the selection of a test to the preparation of the TPS reports. The 2 first steps have been explained in videos already published on Twitter, and the first step, selection of a test, will be the subject of this webinar. The following step will be presented during the next webinars. First, I would like to remind you the definition related to methods and tests. A test is the application of a method to a specific test and a specific matrix. Methods include different methods, like, for example, biochemical methods, molecular methods, or serological methods. A test performance study is the evaluation of the performance of one or more tests by two or more laboratories using defined samples. A TPS is also referred as ring test or collaborative trials. Test performance study is part of validation studies and usually follows preliminary studies conducted in-house. These definitions are part of the EPO standard PM 776. So the selection of tests is critical to obtain relevant results in TPS. Therefore, a set of common rules was prepared in the framework of VALITEST project to ensure transparent process for the selection of the test for TPS. As a reminder, VALITEST project aims at producing validation data for the detection and identification of plant pests. Two rounds of test performance studies have been organized. The first round of TPS was organized in 2019 and targeted 6 pests identified as important by the consortium before the beginning of the project. For the second round of the TPS that was organized in 2020, the pests were selected based on the needs expressed by various stakeholders in order to better align validation priorities with stakeholders' needs and with the market. So the common rules defined in the framework of VALITEST project are the following. First, definition of TPS scope and collection of available data. Then, criteria to be reached by a test. After the analysis of available data that allows the selection of tests for preliminary studies. And finally, preliminary studies according to the criteria and the selection of the final test for the TPS. So first, I will present you the definition of TPS scope and collection of available data. The first step corresponds to the definition of TPS scope, which corresponds to detection and or identification of a pest in a matrix by a method. So it is necessary to prepare a list of known diagnostic methods. The methods differ in their applicability for detection or identification and between the uses on symptomatic or asymptomatic material. In the frame of the scope definitions methods, selection could also depend on the possibility of cross-reaction, on the EU pest status, on the availability of the plant material, and also on the expertise of the TPS organizer. The table presented here corresponds to the list of methods identified by the collection of available data for the TPS Round 1 here in green and TPS Round 2 here in blue in the table. The collection of available data will be detailed in the next part of the webinar. In the framework of this TPS, a wide variety of methods have been selected. So as you can see, there were molecular methods, serological methods, and also other methods like tissue print immunoassay and plating. The next step corresponds to the definition of TPS scope for each method. It is necessary to define the sample type, the matrix, which for the TPS is suitable, the purpose, and the type of controls needed. It is also important to define the number of samples and the number of participants, but that will be detailed in the two following webinars, selection of participants and dispatch of samples. In the framework of TPS Round 1 and 2, we can notice that a wide diversity of samples and matrices have been defined. For example, we can see for sample type, plant extract, wood, leaf, seed, fruits, freeze-dried extract, infected, non-infected plant material, plant material with deactivated pathogen, DNA and rRNA, extract, culture, and tissue print. And for matrices, there are leaves, fruit, freeze-dried leaves and fruits, wood, seed, shoots, stem material, reference culture. So if you want to have more information on the scope definitions for TPS Rune 1 and 2, you can find them in the deliverable D1.1, minimum performance parameters to select tests for validation and selection of laboratories for TPS, and in the deliverable D1.3, list of tests for validation Rune 2 that have been published on VALITEST website, and you have the link here below this page. So, as an example, I will detail now the definition of the scope for the TPS organized on Bursaphenancus xylophilus. Bursaphenancus xylophilus is a causal agent of a pine wilt disease, which may express wilting symptoms in hot and dry conditions, but may also remain asymptomatic in colder conditions. Bursaphenancus xylophilus is a regulated quarantine pest, which implies that any diagnostic test must be highly specific and sensitive. The test should be applied to symptomatic and asymptomatic material. Nevertheless, it is difficult to produce infected wood in large quantity, and it is risky to send such material across EU. Consequently, the biological material is composed either of wood extract spiked with nematodes, either of DNA extract from culture. Therefore, the definition of a scope is molecular detection and identification of Bursaphenancus xylophilus in asymptomatic plant material. And the list of known methods defined for this scope is real-time PCR and LAMP for detection and conventional PCR for identification. Before presenting you the collection of available data, I will now ask you to answer the following question. This is a question that you will have to answer online. According to you, what are the main sources to get access to relevant information on methods and tests available for the pest studies? You have to select maximum 2 answers among research articles, international standards, EPO database, Fresco final reports, and kit providers' websites.
So we have about 75% of the people that have answered. I will wait a few more seconds. It's not changing so much. Yes, so we are at 79%, 80% of the people, and it's not changing so much. So I will close the poll and share the results.
So we can see that EPO, I don't see very well because it's very small screen, but I think it's EPO database who is a majority, and
Yes,
then
it's
research articles and international standards.
yeah.
Okay, so now I will. Okay, I think I. Now we are going to see some interviews of TPS organizers in order to share with you their experience regarding the main sources to get access to relevant information on methods and tests.
how did you get access to relevant information on methods and tests available for the pest studied?
We have selected data on different tests through literature search. We have consulted various databases, including the EPO validation database. We have contacted producers to gather more information, and in some cases also authors of different publications for additional data that was maybe missing in the publication.
As the TPS was dedicated to on-site testing, we consulted the company's websites to find out what kits are available. The information was supplemented by literature search and direct contacts with companies.
One of the first steps that we took when we started our TPS exercise was to consult with a forest plant pathologist, because we needed some general information regarding the pathogen, that it was not really a pathogen that we were used to work with. So we had this conversation just to, as I say, to be put in updated, to be updated on the situation of the pathogen in our area, on the situation of the pathogen in Europe, on the literature that we could have found by ourselves. But of course, if you have a specialist that suggests papers and what to read, for sure it's of a great help. So this is the way in which we started to approach the information regarding Griffonectra parasitica, and after that we did a literature search, as I suppose every other TPS organizer did.
We started with the methods that were in the EPO protocol, and we also used methods from a previous Eufresco test performance study that we'd organised for the same pathogen. And then we carried out a literature review to find any additional published methods. So for this test performance study, we found an additional conventional PCR and 2 additional real-time PCR methods through literature searches. And there was also a survey organised by VALITEST of European labs to see what they were commonly using for the pathogens for the first round of the test performance study. And then we did a search for commercially available kits as well. But for Fusarium circinatum, we found there were no commercial kits available.
I'm really sorry because I think Anna got disconnected.
Do you hear me?
Yes, now we hear you, yes.
Okay. So I don't know if participants can see my presentation. I don't think so. Because we all we see the video still, I think.
Yeah, I will make you the presenter again. Oh, but, oh yes, you are here.
Yeah, I think it's okay. It's okay now. And so as mentioned by the TPS organizer during these interviews, the collection of available data has been realized from research articles, international standards, as for example EPPO and EPPC, EPO database, Fresco final reports, kits provider website, and also an EPO dedicated survey for the VALITEST project. So the table here presented on this slide corresponds to the number of tests identified by the collection of available data for the TPS round 1 and 2. We can notice a large range of identified tests, from 7 to 46 tests for TPS round 1 and from 3 to 76 tests for TPS round 2. As an example, for Bursaphelicus xylophilus, the literature review underlined numerous molecular tests available. 23 publications were identified, including 14 mentioning a conventional PCR test, 5 mentioning a real-time PCR, including a commercial kit, and 4 mentioning a LAMP test. So I'm going to present you now the criteria to be reached by a test to be selected. The criteria were set to objectively select appropriate tests for the defined scope for a specific pest according to the use of the EPO standard PM798, specific requirements for laboratories preparing accreditation for a plant pest diagnostic activity, and from the experience of TPS organizers. For each criterion, there is a descriptor, either quantitative or qualitative, a target to be reached by a test, a relative weight different for the different use of the test, laboratory use or on-site use, and a conclusion whether the criterion is met by the test. So different performance criteria have been taken into account. Analytical sensitivity, analytical specificity, selectivity, repeatability, and reproducibility. So these performance criteria have been detailed in a previous webinar, which the name is What is Test Validation? So I will not detail here the different criteria. And it is important to note that in the selection process of a test, if a criterion is not relevant for a specific method pest combination, it can be ignored. So the table presented here on this slide corresponds to the table that has been used by TPS organizers to collect the data regarding the performance criteria. We can see that for each performance criterion, a descriptor has been defined. For example, for the criterion repeatability, the descriptor is the level, the target is 100% at the level of detection, and the relative weight is medium for laboratory use and on-site use. So it has to be noted that criteria can be differently weighted to allow the selection of appropriate tests for the defined scope for a specific pest. And some additional information can also be indicated, but it is not considered as a criterion. So if some of the tests show similar value and performance, then also less important criteria can be taken into account: applicability, protocols, chemicals, and equipment. These other criteria help to evaluate other properties of specific tests. It is important to note also that if a criterion is not relevant for a specific method pest combination, it can be ignored. So here is a table that has been used by TPS organizers to collect the data regarding the other criteria: applicability, protocols, chemicals, and equipment. For example, for the criterion applicability in different matrices, the descriptor is the level, the target is high, and the relative weight is medium for laboratory use and on-site use. Criteria can be differently weighted to allow the selection of appropriate tests. And I will not detail here all these criteria during this presentation, but if you want to see the complete tables criteria for the selection of tests for TPS round 1 and 2, then you can find them in the deliverables D1.1 and D1.3 that have been published on VALITEST website. So the third step corresponds to the analysis of available data in order to select the tests that will be subjected to preliminary studies. The analysis of available data corresponds to the analysis of performance values from the available validation data for each test and to the objective comparison of performance among the tests identified. So we are now going to see some interviews of TPS organizers in order to share with you their experience regarding the difficulties they encountered to select the test and how did they circumvent this difficulty.
What was the major difficulty you encountered to select the test, and how did you circumvent this difficulty?
First, I would say that it was quite obvious from the start that a lot of effort goes into generating data on the test performance, but it does also reflect the richness of plant health area. Everybody is doing things slightly differently, using a bit different reagents, using different instruments, of course. And while I would say that this is really good for the plant health area because it means there are more lines of defense against introducing a pathogen. If I'm not detecting it 100%, somebody else will catch a bit deviant isolates. It does have a practical consequence that sometimes it's easier to redo validation than actually collect the data. And even when you go through this time-consuming process of collecting data, it is often not really comparable. So although we have made huge advances, I would say, particularly with the validation database of the European Plant Protection Organization, now redesigned to make it even more structured, I would say that there's still some space to improve this. Not only at the level of exchange of data among the laboratories, but also within the laboratories. And I think the next big thing is generating and collecting data in line with fair data principles to make it more easily findable, accessible, interoperable, and reusable.
As there are only a few on-site tests, the first selection was easy. Abnormal results were obtained for 2 out of 3 tests. Since the matrix was suspected to be the cause, corrective adjustments had to be made.
So for the molecular test, we did not really encounter any major issues. For the serological test, the main difficulty was the selection of the actual tests. And this was because there was very little validation data available. So we really didn't have anything to go on. And furthermore, not all of the kits were available at the time that we wanted them. So that made it difficult to actually select a test that we would use. So in the end, we ordered the kits that we could get our hands on, and we tested those. And we selected the most sensitive ones. Although one of the difficulties we had, and this is also question 7, is that we followed the APO standard PM7-125 ELISA test for viruses. And this is also part of our standard operating procedure in the lab. In some cases, this standard deviates from what the manufacturer protocols say, but we followed our own protocol. So in that sense, the selection of the serological test was not ideal because we did not follow the protocol of the manufacturer. So in that sense, we may not have selected the best test for the performance study.
So as mentioned by TPS organizer on the interviews, the main difficulties encountered for the selection of a test are for some tests extensive validation data, and for some tests lack of validation data or no validation data. That implies a very difficult comparison. The other difficulties are specific equipment required for some tests, availability of commercial kits and or reagent, and some tests that are not widely used in diagnostic laboratories in EU, meaning that no EU research publication and that they are not included in EPPO and EPPC diagnostic protocols. So as an example, for Bursaphenancus xylophilus, the analysis of available data has been realized from the availability of validation data from literature sources, from tests reported in EPPO and EPPC diagnostic protocols, from tests reported in EPPO survey results, from available results of inter-laboratory studies or intra-laboratory validation data, and of course from the experience of the TPS organizer. This analysis allowed the selection of 6 tests to be subjected to preliminary studies. So I will detail here the 6 pre-selected tests. So literature review underlined a large offer of tests, and only tests corresponding to the TPS scope focused on high analytical sensitivity and high analytical specificity have been selected for preliminary studies. In particular, the test Bürgermeister is widely used in the EU region and is considered as gold standard. And for real-time PCR, only one commercial kit is available and has not been previously submitted to an interlaboratory study. The LAMP method has been also selected for this TPS, as on-site testing is crucial for early detection, and the tests have been selected to document their performance. The 2 tests selected among the 4 identified in literature have been selected based on the results of EPO survey. So just to show you how TPS organizer did practically this selection. So here you have a table, and it has been realized by depending on the availability of validation data and based on comparison of performance. So these 2 points have been taken into account to objectively select the test for preliminary studies. For example, for the method conventional PCR, the test Bürgermeister has been selected for preliminary studies due to its good validation results and due to the fact that it is a test widely used in EU laboratories. The test LEAL has not been selected because it is a test that is less efficient regarding specificity, and the test LI has not been selected due to the absence of validation data. So the table presented here corresponds to the number of tests selected for preliminary studies for TPS Rune 1 and 2. It has to be noted that a special care has to be taken regarding time and budget constraints to perform preliminary studies. So the final step corresponds to the preliminary studies according to the criteria defined earlier and the selection of a final test. So as an example, for Bursaphenancus xylophilus, we have seen already that 6 tests have been selected for preliminary studies. So if you want to have more information on the selected tests for preliminary studies for TPS Rune 1 and 2, again, you can find them in the deliverable D1.2 and D1.3 that have been published on VALITEST website. So here, as an example, you can see how have been selected the tests for preliminary studies. For Bursaphenacus xylophilus, preliminary studies included analytical sensitivity, analytical specificity, repeatability, and reproducibility. Performance criteria have been taken into account to objectively select the tests for the TPS as already detailed in the part criteria to be reached by a test. In this table, we can see as an example the performance value for the test Burgermeister for the conventional PCR method. So the analysis of performance values has been realized for each test in order to select a final test for the TPS Bursaphenacus xylophilus. For real-time PCR, only one commercial kit, as I already said, clear detection was identified for the detection of Bursaphenacus xylophilus and is available on the market. Moreover, preliminary studies gave correct validation results. Therefore, this test has been integrated to the TPS. Among the two LAMP tests evaluated, the test developed by Meng and al. Did not give any result and was consequently not included in the TPS. Preliminary studies of the other preselected tests gave good results, and then these tests were therefore included in the TPS. Consequently, 5 tests have been selected for the TPS on Bursaphenacus xylophilus. So for information, the test Kikuchi and for the method LAMP and the test clear detection for the method real-time PCR will be added to the revision of EPRO protocol for Bursaphenacus xylophilus. In order to select the test for the TPS, we have seen during this presentation that it is necessary to follow different steps. Definition of TPS scope and collection of available data, analysis of available data and selection of tests for preliminary studies according to the criteria that have been defined, and finally preliminary studies according to the criteria and the selection of the final test. So as an example for Bursaphenacus xylophilus, we have seen that 3 methods and 23 tests have been identified from literature, 6 tests have been selected for preliminary studies, and 5 tests have been finally selected for the TPS. Therefore, the selection of the test, as we can see, is a long process from the identification of method and test to the final selection of tests for the TPS. So I will now ask you to answer the following question. According to you, how long does it take to perform test selection, including preliminary study? Less than 3 months, between 3 and 6 months, or more than 6 months?
So we are almost at 80% of the people that have voted. I will wait a few more seconds. Okay, so now it's 86%. It's not changing so much, so I will close the poll and share the results. So
yeah, so we can see that yes, we can see that you think that it's between 3 and 6 months for the process of the selection of the test. So we are now going to see some interviews of TPS organizers in order to share with you their experience regarding the time necessary to perform the selection of the test.
Yes, so for this video, I am very sorry, but before the start of the meeting, we got some trouble to upload the video. So actually, I will just share the video on my screen, but it means that you won't be able to hear what the TPS organizer are going to say. So for the one of you that are on their computer, I will also put a link, a YouTube link to the video on the chat. So you can probably, it's better if you just open the link and watch the video on YouTube. And I will also start the video on my computer. Even if you cannot hear, you will see the response that the TPS organizers gave. So now we can go back. Sorry again for this little glitch. So I hope you were able to see the videos on the share screen or on YouTube. So now I will give back the floor to Anna.
Okay, so as mentioned by TPS organizer, I don't manage to. Okay. So as mentioned by the TPS organizer during the interview, so 5 TPS organizer mentioned that it is necessary to take 2 or 3 months for the selection of the test. For 1 TPS organizer, it corresponds to 4 to 5 months, and for 3 TPS organizers, it corresponds to 5 to 8 months. So it's important to note that the number of staff involved in the TPS organization team and the number of selected tests for preliminary studies have an influence on the timeline. So to summarize, the table presented on this slide corresponds to the different steps regarding the selection of the test for TPS round 1 and 2. So first, identification of methods and tests by the collection of available data. Then the selection of the tests subjected to preliminary studies according to the defined criteria. And finally, the selection of the final test after preliminary studies. So we are now going to see some interviews of TPS organizers in order to share with you their experience regarding the difficulties they encounter to select the test.
Did you encounter unexpected difficulties?
Yes, this for sure was that we did not expect, for example, that each commercial ELISA kit required different buffers. Our lab is small, so the first thing not easy to be solved is where to store all bottles with all those different buffers. But a more important question related to these differences between commercial kits we had were how to prepare a now plant material, and even more difficult question we had was how to prepare homogeneous plant material for comparison study.
Yes, we did, because as explained previously, in some cases we had to adapt the protocols because reagents mentioned in the scientific publication were no more commercially available. So we had to find new reagents which can be used for the protocols.
The main difficulty that we encountered was the high concentration in brown rugus in leaves and fruit that poses high risk of contamination during the manipulation in laboratory. Extreme care must be taken in all the steps to avoid cross-contamination and false positive results. Moreover, all the real-time RT-PCR tests highlight slightly non-specific emission in fluorescence at late GQs that bring problems in the interpretation of results.
Yes, one of the unexpected difficulties we found was that the primer and probe concentration in the EPO protocol differed from the published paper, along with the magnesium concentration for one of the real-time PCR assays. So we had to do some work to look at whether the paper concentrations or the EPO protocol worked more optimally for that assay. And we did find that that assay was particularly susceptible to changes in primer concentration and different master mixes being used. So that had to be explained in the technical sheet that went out for the test performance study, so that participants were aware that it was quite important that they used a particular concentration for that assay.
So as mentioned by the TPS organizers during the interviews, some difficulties have been encountered during the process of selection of a test, and some lessons have been learned from TPS organizers' experience. So it is not possible to include all available methods and test kits, in particular where the number of tests available was very high due to time and budget constraints. Sometimes the absence of critical information can limit the inclusion of the test into the TPS validation procedure. In some cases, no commercial kits are available on the market, or TPS organizer is not aware of its availability. And in some cases, if the offer of kits is too large, it is difficult to validate kits in their original description. For TPS Round 1, the preselection of the test has been realized from the expertise of TPS organizers, previous experience, and assessment of feasibility. For TPS Round 2, the preselection has been based on input of kit providers, especially with regards to modifications. So if the TPS organizer has a lack of expertise on a method, then the method will not be included in the TPS. As TPS organizer mentioned during the interviews, the protocols sometimes needed to be adapted. So the most common reasons was adaptation of sample preparation, including the extraction buffer to a particular matrix, the replacement of reagents specified in the original source but no longer available, the use of different instruments, the optimization of processes, as for example, minimizing the number of different buffers, polymerases, instruments, DNA extraction kits in a laboratory. So regarding the selection of a commercial kit versus a publication, that will depend on validation data for commercial kit compared to the same test from publication. Therefore, a more transparent information on validation data and performance is required for commercial kits inclusion in TPS. So as a conclusion, discussions and harmonization between TPS organizers and commercial kits providers is essential in the process of the selection of a test. So thank you for your attention, and if you have any question, please ask them either by chat or raising your hand. Thank you.
So I will also ask the TPS organizer to join us for the Q&A session. So Marta, Anne-Marie, and Tanya, if you can, turn on your camera. And so, do not hesitate to ask the questions to the TPS organizers in the chat, or you can also raise your hand if you want to take the floor. So there was one question from Dina, which was why qPCR is used for detection and conventional PCR used for identification. So I guess it depends. Maybe one of the TPS organizer can answer this.
Yeah, I think that if Anne-Marie Chappé, I think explained that detection is on wood extract, meaning that we don't know if there will have some nematodes, so that will be detection. And for identification, we know that there are some nematodes, but we want to know if it will be the one we want to identify. So for example, for Bursaphenacris xylophilus, we want to identify if it's really the Bursaphenacris xylophilus. I think it's that. Yes.
I think that what we can say, because I don't know if the question was specific for the case of Burstaphylancus or more in general,
not, yeah.
but in general, for detection, we prefer to have tests that are more sensitive. So that's why qPCR, real-time PCR, might be preferred for detection than and conventional PCR might be more specific. So sometimes they are used more for identification, but I think it's not something that is general. It really depends on the pest and on the protocol. Marta, maybe you want to add something?
Probably also because the conventional PCR could be sequenced, so you can be really sure about the identity of the target that you are looking for. Probably also this could be these differences.
So there is another question. I think it's for all TPS organizer. Did you easily retrieve information on the performance of the test, including CT, well, and I guess a comparison in literature? So I don't know. I think we got some answers in the interviews, but maybe Marta first, if you want to, and then Tanya, yeah.
We organized the TPS for tomato brown rugose fruit virus, and all the protocols are really new. Were really new, so we don't have a lot of information on the publication about the performance of test and also on the CT study. And we found that we had some problem at the high CT. No, we don't have any information about comparative studies for tomato brown rugose.
Maybe Tanya, you want to share your experience?
Yes, thank you. I would say that this part is probably the most frustrating, if not the most difficult. And basically, you just have to collect the different sources of information and really read them, often in details, trying to find all the small pieces of information that may be important. So in that respect, I would say that we are quite far from making this easy. Ultimately, you can imagine that there is plenty of validation data that is sitting in the lab with all the small modifications they have done and is not shared, often simply because of the constraints of time. People do not have time to put this data into a form that could be easily exchanged and ultimately machine readable, so that you could really pull it together in a simplified, easy way.
So there is another question. Could you clarify how the data was collected from the participant laboratory? Was it using a survey? I think it's a bit out of scope, but maybe one of you can give some information.
Is this about data on validation data on the tests or the results of in the test performance study?
Maybe Lina, you can clarify. She said the results of the TPS.
Okay. Well, very briefly, I think this will be addressed in some later webinars, I guess. But in our case, at least, we have prepared Excel sheets to collect the data as structured as possible. And within these Excel sheets, we had some formulas and Power Queries behind that have translated this data that participants have entered into the real sample numbers, because the samples, for example, in our TPS studies were also randomized. So sample number 1 was not the same for each of the participants. And we had to translate these numbers and pull them together. So yeah, that's basically it. Excel sheets with all the structured data.
Okay. And there is one last question. Is qPCR sensitive enough when a virus is present in low load? Will be sequencing a solution in this case? Maybe Marta, you can.
Depending on the performance criteria of the test, if the qPCR is sensitive enough. In our case, we have so much amount of brown rugose in samples that we didn't have any problem in sensitive. But for my experience, it could happen. Generally, if a real-time PCR is not enough sensitive, you cannot sequence an amplification, because generally the conventional PCR is less sensitive than real-time RT-PCR. So I don't think it could be a solution.
Sorry,
Yes.
Charlotte, if I may add, I agree with Marta. But maybe I would just like to share our experience that at least in bacteriology, sometimes classical PCR has the same analytical sensitivity as real-time PCR. Not often, but it happens. At least in our case, this is for potato quarantine pathogenic bacteria, Ralstonia and Clavibacter. However, at the same time, I would say that with the tests themselves, we are already very close to their theoretical sensitivity, and we are no longer limited by the sensitivity of the test, but simply by the amount of sample that we manage to put into the reaction. So if you can imagine potato testing, we start with 200 tubers, which is, you know, a big bag of potato tubers, and we prepare from all of this 1 ml extract. We take 100 microliter, put it into the DNA extraction, and then finally, in the end, we put maybe 2 microliters into one real-time PCR reaction. Even if we do 3, we end up with 6 microliters out of that big bag of potatoes. So this is the main bottleneck that is still remains, and we haven't solved it, and probably it will still take some time to solve it. With regards to sequencing, it's a very similar problem. So you either have to sequence very deep, in the case of HTS, to get the sensitivity, which again is still quite expensive, or alternatively, or better still, in combination with that, you want to put more of the sample into the reaction to basically get the target into what you are putting into the sequencing.
So I can see no more question, and I think we have passed the time. So I would like to thank Anna for the presentation, and Marta, Tanya, and Anne-Marie for being there for the Q&A session. I would like to thank all the participants for joining the webinar today. There will be a satisfactory survey at the end of the webinar and in the follow-up email. So it is really short, so do not hesitate to provide feedback to us to help us improve the webinar experience. And also, if you have some question, you can ask them there. The presentation and the link to the recording will be on the website. And Anna, if you can change to the last slide. So the next webinar will be next Monday, and it will be on the selection of the participants and on the contract. So do not hesitate to register for this webinar as well. And thank you all, and see you next Monday.
Okay. Thank you.